Good morning and welcome, everyone. Thank you for joining us for the Chabner Colloquium MAP series. My name is Dr. Ryan Sullivan from the Mass General Hospital Cancer Center, and I will serve as a moderator for today's session, which includes presentations by Dr. Justin Gainor and Dr. David Spriggs. Before we begin, I'd like to thank our sponsors, including Novartis Pharmaceuticals and Regeneron Pharmaceuticals and Sanofi Genzyme for providing educational grant support for this series. I also want to acknowledge Dr. Aditya Bardia, the MAP series co-chair, and Massachusetts General Hospital for collaborating with Dr. Chabner and the STO on the development of this series. We are especially grateful to the presenters for their participation. Additionally, I encourage you to visit our exhibit hall, which you can access in the timeline on the main event page to learn more about each of our generous supporters. They include AstraZeneca, Foundation Medicine, Genentech, Novartis, Pfizer, and Takeda. They have posted informational brochures and videos for you to download and view at your discretion. In addition, you may set up a chat with a representative if you desire. Information was emailed to attendees with details about how to receive continuing education credit for this event. And now I would like to introduce Dr. Gainor, who will be presenting on the role of immunotherapy in the era of precision medicine, and Dr. Spriggs, who will discuss the role of antibody-drug conjugates in the era of precision medicine. Immediately following both presentations, we will host a 15 to 20-minute Q&A session. If you'd like to submit a question to be addressed, please use the chat function of this platform to post your inquiry. Questions can be submitted anytime during the presentation. Please note that the microphones for all attendees are automatically muted. Without any further comments let us proceed with today's session.
Hello, my name is Dr. Justin Gainor from the Massachusetts General Hospital, and it's my pleasure today to speak to you on the role of immunotherapy in the era of precision medicine. I'd first like to thank the Society of Translational Oncology, as well as our chairs, Dr. Bardia and Dr. Chabner, for the invitation to speak to you today. Here are my disclosures. In terms of learning objectives, I've identified three. That is to identify emerging strategies for use of cancer immunotherapy in solid tumor oncology. Two, to summarize the clinical applications and limitations of predictive biomarkers of immune checkpoint inhibitors. And three, develop a translational framework for evaluating additional IO biomarkers. It's important as we delve into this topic to first acknowledge that cancer immunotherapy encompasses a very broad spectrum of therapies, ranging from cellular therapies to checkpoint inhibitors to cytokine therapies, vaccines, and monoclonal antibodies. So this is really a massive topic, and depending on the disease of interest, we may be focusing on one or several of these different therapeutic modalities. I'm going to be focusing my talk, however, on immune checkpoint inhibitors, as really because these are agents that have transformed the management of solid tumor oncology. And we're really going to be focusing on two in particular, CTLA-4 inhibitors and PD-1, PD-L1 inhibitors. And just to really emphasize how much these agents have transformed cancer care, I'd like to take a lesson out of melanoma. And the figure on the left, we see this is a meta-analysis of 42 phase 2 clinical trials conducted in melanoma between 1975 and 2005. And we can see the Kaplan-Meier curve here on the left. And now, if we put on the right a Kaplan-Meier curve for the 5-year survival outcomes of the PD-1 inhibitor pembrolizumab, we can see that the tail of this curve has been lifted, where we're now seeing 5-year survivals of more than a third of patients. So this really speaks to the transformative nature of immune checkpoint inhibitors. And this is a story that's been played out in multiple disease areas. Indeed, over the last several years, we now have checkpoint inhibitors approved in more than 15 different indications. And this is a figure that is constantly evolving with a growing number of approvals, including approvals in tumor agnostic indications, such as microsatellite instability or tumor mutation burden, as well as approvals moving into earlier disease setting, such as the adjuvant setting. Given the purposes of our time here today, I thought it best to focus on one particular disease area, as really this encompasses many of the principles that we're seeing of cancer immunotherapy. And for that, I've focused on non-small cell lung cancer. Here I've depicted the various regulatory approvals in non-small cell lung cancer of various checkpoint inhibitors over the last 6 years, and you can really see a flurry of different approvals. If we though break these down, these really go into three big strategies. The first strategy being use of PD-1 inhibitor monotherapy in a biomarker selected patient population. In the upper right, another strategy is to use chemotherapy together with checkpoint inhibitors. And then below, a third strategy is to use checkpoint inhibitors in combination with one another, leaving out chemotherapy. And so all three of these strategies have now been deployed. All three of these strategies have regulatory approvals, but how do we make sense of them? How do we actually deploy them in a precision way? So this is a perfect segue to our discussion of biomarkers. I thought I'd focus this talk on biomarkers because it's really biomarkers that allow us to pursue precision medicine. And here, in contradistinction to some of the other talks that you've already seen in this series of targeted therapies, cancer immunotherapy has a unique challenge in that we're not just focusing on the tumor, but we're also thinking about the tumor microenvironment. And there can be things within the tumor microenvironment that confer resistance to our therapies. And so if we actually take a step back and think about the determinants of response to checkpoint inhibitors, they're actually quite broad. We can think about host factors. So these can range from a microbiome to germline genetics, to tumor intrinsic factors, and we're going to touch on many of these, to things in the tumor microenvironment, to other clinical factors that may be influencing how a particular patient responds to therapy. I'll begin first, though, with PD-L1 expression. This is the biomarker that's been most studied in non-small cell lung cancer. And a few key points here. First, if we look on the left-hand side, this is in PD-L1 unselected populations. We see that the response rates to checkpoint inhibitors range around 20%. If we then look at PD-L1 selected populations, and here I've provided various cutoffs from various pivotal studies, we can see an incremental increase in response rates as we go up in PD-L1 scoring. However, we see that most patients still don't respond to PD-1 inhibitors or PD-L1 inhibitors, even when we're at our highest cutoff of 50% shown here. And so when I think about PD-L1 expression as a biomarker in lung cancer, I'm commonly reminded of this quote attributed to Winston Churchill, that no one pretends that democracy is perfect or all wise. Indeed, it has been said that democracy is the worst form of government, except for all those other forms that have been tried from time to time. And so instead of democracy here, I would actually plug in PD-L1 expression, whereas it is very much an imperfect biomarker. But what we've seen in non-small cell lung cancer is that it still remains the cornerstone of decision-making when it comes to cancer immunotherapy. And so far, we haven't seen a biomarker that really displaces it. And so how do we actually use it? So I've shown here two pivotal studies looking at, again, PD-1 monotherapy in PD-L1 selected patient populations. On the left is Keynote 24, And this used a cutoff of 50%. So patients with PD-L1 expression in their tumor cells of 50% or more were randomized to either pembrolizumab or chemotherapy. And you can see here a significant improvement in overall survival among patients receiving pembrolizumab. By contrast, if we look at a very similarly designed study, this was Keynote 42. And in fact, this study actually looked at 50% and above, as well as the 1 to 49%. I didn't show the 50% and above, but it pretty much recapitulates the findings on the left. That is, there was an improvement with pembrolizumab among patients with 50% and above. But if you look at the 1 to 49% subgroup, we see that the curves cross and this is no longer significant. And so this has created 50% PD-L1 expression is a magic number in thoracic oncology, and that above that number, we commonly will pursue PD-1 inhibitor monotherapy. I introduced that there were actually three major strategies for using checkpoint inhibitors in lung cancer. Monotherapy was just one, but chemotherapy combinations were a second strategy. And here I've summarized a lot of data in one table, but essentially here we see 6 different randomized studies evaluating typical platinum doublet chemotherapy versus platinum doublet chemotherapy with the addition of a checkpoint inhibitor. And you can see across all 6 of these studies, there was an improvement in progression-free survival with the addition of checkpoint inhibitor, and 4 out of the 6 studies had improvements in overall survival. And really, this has fundamentally changed the standard of care for non-small cell lung cancer. Specifically for patients who don't have high levels of PD-L1 expression, the standard is really now chemotherapy plus checkpoint inhibitor. And this is just to illustrate the point that these benefits of chemotherapy plus checkpoint inhibitor are seen across the spectrum of PD-L1 expression levels, including patients who have 0 PD-L1 expression. And so this has really culminated in a treatment algorithm that looks like this. I apologize. I know this is a ton of information to pack into one slide, but to take you through it very broadly, if we start with non-squamous histology, we need to do genotyping to test for targetable alterations as well as PD-L1 testing. If patients have an oncogenic driver, which again, you've heard about many of these during this series, the standard of care is targeted therapy. Beyond that, if someone doesn't have a targetable driver, we look at PD-L1 status. And depending on that, we can use one of the 3 different strategies that we talked about: PD-1 monotherapy, chemo plus PD-1, or IO combinations. In practice, the most common strategies have been high PD-L1 expression. We generally give PD-1 monotherapy. PD-L1 expression less than 50%, we're most commonly giving chemotherapy plus PD-1, but there are many other approved regimens, as you'll see depicted below. So that is currently the state of the field, that we're really only using PD-L1 expression as our biomarker. But as you've seen for the targeted therapy world, the outcomes aren't good enough and don't really replicate what we see in the targeted therapy world in terms of response rates. And so clearly there's a lot more to understand here. And the goal is to actually add in additional biomarkers to help us distinguish who can just have monotherapy, who actually needs some sort of combination therapy. And so today I was going to just highlight 4 different biomarkers and then close by talking about how we can actually get additional clinical data to validate some of these. So the 4 I'm going to focus on are tumor mutation burden, several single gene predictors, smoking status, and CD8 tumor infiltrating lymphocytes. So first, tumor mutation burden. So this is really a a biomarker that most will be familiar with. This is really seen as a surrogate for neoantigen generation. And this is a figure of a really pivotal study from Nair Rizvi and Matt Hellman, published in 2013, where they took a series of about 32 lung cancer patients. They did whole exome sequencing, and they observed that patients who had a higher tumor mutation burden were more likely to have durable clinical benefit following treatment with checkpoint inhibitors. So this is a biomarker that's commonly reported in sequencing assays. It's been incorporated into multiple clinical trials. But right now, the clinical utility of tumor mutation burden, or TMB, in lung cancer really is it's still looking for a role. And that is because it's both prognostic and predictive. Still question marks regarding the optimal cut point. Do you focus on tumor? Do you focus on blood? And really the pivotal phase 3 studies really haven't played out with TMB as a biomarker. So it really hasn't come into the forefront of clinical use right now. And so there are still ongoing efforts to try to further refine TMB. In terms of single gene predictors, one of the strongest single gene predictors is STK11 or LKB1, and this is actually a marker of resistance. So STK11 is commonly co-mutated with KRAS. On the left is data from a stand up to cancer mutation, a stand up to cancer consortium that we were fortunate enough to participate in. And it was really looking at patients with KRAS mutations. So those are depicted K only in green, or KRAS plus P53 mutations in blue, or KRAS plus STK11 or LKB1 in red. And what you can see is that the response rate to checkpoint inhibitors among patients in the KL group was significantly lower than the other KRAS subgroups. So it seemed like LKB1 was modulating responsiveness to checkpoint inhibitors. And then in panel B is actually data from a prospective study, Checkmate 057, where this was also investigated. And we can see that the response rate among the KL group is also low. And on the right, this is actually knockout data showing that so in STK11 knockout mice, you actually are no longer responsive to checkpoint inhibitors. So this is actually one of the strongest single gene predictors, and we're still working on identifying the mechanism that's driving this. Similarly, among other oncogenic drivers, EGFR mutations and ALK rearrangements also confer intrinsic resistance. In the upper left-hand corner here, this is data from Mass General, where we showed that these patients have a much lower response rate compared to wild-type patients. And this has now been borne out in meta-analyses. So in the upper right, this is a meta-analysis showing that EGFR patients don't derive as much benefit to checkpoint inhibitor compared to wild-type patients. And then below is in a prospective clinical trial that specifically enrolled patients with EGFR or ALK. We could see the response rates were quite low. The response rate was 3.6% among patients who had low PD-L1 expression, and was only 12% among patients with EGFR or ALK who had high expression. And this is not just unique to EGFR and ALK. This is taken from the Immunotarget Registry, which was looking at various oncogenic drivers. And we can see that in addition to EGFR and ALK, RET, ROS1, HER2, these are patients who have relatively low response to checkpoints, as shown here. And what could be the mechanism that's driving this? So likely several things. First, we've learned that PD-L1 expression is really not a good biomarker specifically for these oncogenic drivers. And that's because PD-L1 expression can be upregulated for several reasons. In the figure here, in the upper left is adaptive immune resistance. So commonly PD-L1 is upregulated in the setting of interfering gamma that is being secreted by infiltrating immune cells. By contrast, though, PD-L1 can also be upregulated in the setting of just oncogenic signaling, which is depicted on the right, so-called innate immune resistance. So that is, PD-L1 is present despite infiltrating immune cells. And indeed, when we look at EGFR or ALK positive patients, we see that PD-L1 expression can be upregulated in 11 to 26% of patients, as shown in this series, but very few have concomitant infiltration of CD8 positive infiltrating lymphocytes. So this really, we think is an important piece in that PD-L1 can be a red herring, particularly in these oncogenic drivers. And we also think that there's a role of smoking here, and that if you look at tumor mutation burden according to smoking status, never smokers on the left, current smokers are on the far right. And so we see about a tenfold difference between tumor mutation burden between smokers and non-smokers. And so it's likely that patients with EGFR, ALK, and other driver mutations, which are enriched for patients who are never smokers, likely have low tumor mutation burden and generally lack neoantigens, and that's likely driving some of the lack of responsiveness as well. To hone in on the smoking status piece a bit more, this was a series that we looked at. So we decided to focus in on patients who had high PD-L1 expression, so 50% and above. I said, this was the magic number in lung cancer. And we looked at, regardless of driver status, we just looked at smoking status. If you were a never smoker, light smoker, that was up to 10 pack years, or a heavy smoker, more than 10 pack years. And we see that there is a difference in both progression-free survival, which is on the left, that was non-significant, but also the duration of response on the right between heavy smokers and never or light smokers. That is, never or light smokers, we see a trend towards reduced duration of response. Now, given the numbers, this is something that we really need to explore in larger patient populations to try to get better numbers here. In the closing minutes, I just wanted to say a brief word about CD8 tumor-infiltrating lymphocytes. This has been observed as a biomarker of response in multiple tumor types. This is a very busy slide, admittedly, but this was a pan-cancer analysis of both genomic and transcriptomic analysis, looking at over 1,000 different patients. And in the bottom figure, in the bottom portion of the figure, looking at CD8 positive infiltrating lymphocytes, looking at expression, we do see that this enriches for response in this series. So how can we explore additional biomarkers and really sort out what's likely to be a need for combinations of biomarkers? Well, this is one such effort that is taking place. This is a Stand Up to Cancer Mark Foundation extol team, which is comprised of multiple different organizations, but 9 different institutions, where we're taking standard of care biopsies, but subjecting them to whole exome sequencing, RNA sequencing, multiplexed immunofluorescence, and really correlating this with clinical outcomes to checkpoint inhibitors to try to get a better sense of what the best biomarkers are. And we're taking this now into deeper analyses, looking at single cell RNA sequencing, looking at germline data, as well as using peripheral T cell analysis to track neoantigen reactive T cells. And really the goal here is both to develop better biomarkers, but also to inform the next generations of trials. This slide summarizes various clinical trials that are taking place, evaluating different IO combinations, and it's really impossible just empirically pick what the most promising combination is going to be. And so we're going to need to rely on biomarkers to help sort this out. And so to close, I'd like to emphasize that cancer immunotherapy encompasses a broad spectrum of anti-cancer agents. And these checkpoint inhibitors have really transformed the management of solid tumor oncology. But as only a subset of patients respond, efforts are needed to develop more effective combinations. And given the sheer number of possible combinations, it's going to be imperative to develop novel trial designs and strategies and better biomarkers, as well as biomarkers of resistance to help guide identification of the most promising combinations. And with that, I would like to say thank you and look forward to answering your questions.
Good morning, and thank you for joining us today for the Chabner Colloquium. I'm David Spriggs. I am the director of the Medical Gynecologic Oncology Group, and my research is focused on monoclonal antibodies and their development for therapeutics. And I'm going to talk to you a little bit about monoclonal antibodies and the technology maturation that's been ongoing for the last couple of decades. And so I'm looking forward to speaking with you. I do have some conflicts of interest to declare. I'm a consultant for Repertory Immune Medicines, which is a T-cell-based company in Cambridge, Massachusetts. And I own patents through my work at Sloan Kettering and the MGH. Some of those patents are licensed to Eureka and Bristol Myers Squibb in a CAR T-cell development pathway. I also own some patents on galectin-3, but I don't think those will be relevant today. So I'm going to talk a little bit about how antibodies have really assumed a preeminent role in the therapy of cancers, both hematologic cancers and solid tumors, over the past decade or so. And I want to talk about some of the fundamental strategies for antibody therapies and how they differ and what the advances today are showing. So this is a graphic taken out of a review by Jed Wolchuk and his colleagues looking at different ways that monoclonal antibodies can interact with a tumor cell. And in the upper left-hand corner there, you can see that the antibodies can interact with both receptors at the 12 o'clock site and then at cell surface enzymes. They can also be carriers for conjugates, and they can act as an agonist for certain kinds of receptors. They also mediate immune-based tumor cell killing, as shown in the on the right side, with interacting with macrophages to generate phagocytosis, to activate complement, to activate the NK cells going around clockwise, and then even develop modified T-cell receptors using a single chain from a characteristic monoclonal, or and then going on to presentation through the MHC with T-cell interactions that might be delivered through that same strategy. So if one thinks about the antibodies in the most broad version, you've got variable regions for both a heavy chain and a light chain. You have carbohydrate conjugations, and then you have a constant region with an Fc receptor at the other end of the receptor bottom on the screen. And so if we go around again now counterclockwise at some of the possibilities, you see one of the first things that can happen is the antibody can have its glycosylation modified through the production of strategies where they're produced in cells that are deficient in some glycosylation enzymes. You can also alter the amino acids themselves in the constant region to make them more or less stable. You can use different isotypes, and the different isotypes will result in different properties. You can link isotopes to the monoclonal with a stable linker, and you can either do imaging or radioimmunotherapy. You can link to a drug and have the development of antibody drug conjugates, but you can also insert the sequence for the antibody variable region as a single chain into CAR T cells with lentivirus or retrovirus to end up creating CAR T cells. You can also take the variable regions of different antibodies and connect them together. So you can see that already taking a single monoclonal antibody, there are many different therapeutic opportunities that can be created as we go forward. So if we think about how to build those monoclonal-based therapeutics, we come to the following kinds of constructs. One is that you can have a single chain, which is basically the variable regions of the antibodies, heavy chain and light chain, which have been linked together through usually it's about a 25 amino acid linker. And what that gives you is it gives you a small protein, which is ideal for diffusion and imaging. It's not ideal because it has a very short half-life. Most of the single chains end up being filtered by the kidney and excreted very promptly. One way to get around that is to make what's a so-called diabody, where you basically have two single chains connected together, and those are big enough that they're much less likely to be filtered by the kidney and excreted. You can have the other versions, mini bodies, where they're cut and the FC is removed, for example, and you can also have a full protein with an FC that seems to be active and can bind to certain kinds of FC receptors. You can create IgE or IgMs, although that's much less common. And you can go on to create drug conjugates, for example, and then you can label nanoparticles and deliver a variety of things inside the nanoparticle liposome kind of strategy, or develop bispecifics where things like anti-CD3 or anti-CD8 may be used to activate T cells, specifically when the other end of the molecule is connected to a cancer target. There are lots of strategies associated with this, and now you're thinking about what kind of target are you choosing. And this table illustrates some of the targets that can be chosen, including tumor antigens, which are prime targets for antibody-based cell destruction. And you can also just use them as an unmodified antibody to inhibit things like angiogenesis by blocking the receptors. You can block the molecules like PD-1 or CTLA-4, which alter the T cell response. You can deliver radiolabeled like lutetium, for example, which is now being done in certain kinds of GI cancers, particularly in the small cell and carcinoid versions. You can see conjugates where you put a highly toxic target onto the molecule, and again, you're now going back to a highly specific tumor-associated antigen, which internalizes itself and allows the internalization of the drug conjugate. You can also hook it up with the anti-CD3 antibody or create other strategies with T cells. Lots of targets to consider. And if you look at the table on the right, you can see some of the differentiation antigens that are common in hematopoietic cells like CD20, CD33. And you can also look at glycoproteins, so EpCAM, CEA, things like MUC1, MUC16, PMSA, all are logical targets there. You can also do simple carbohydrates like the Lewis Y antigen, or you can think about vascular targets or receptors like EGFR or B3 and so on. So there are a variety of different strategies that can be used, and it's not just targeting the tumor cell itself, it's targeting every aspect of the tumor microenvironment, whether it's a tumor cell, whether it's the fibroblast, the immune system, or the vasculature of that target. All of them are available as as antigenic targets for a monoclonal antibody strategy. There are lots of different mechanisms for antibodies, including antibody-dependent cytotoxicity, complement fixation, and blockage of signal transduction inhibition. And the table on the right there just goes through some of those examples. I want to touch on the other thing that you can do, which is you can really change the behavior of an antibody by changing the glycans on the cell surface of the antibody. This is a paper from Jeff Rabich's group in PNAS in 2017. And what they did here was they looked at rituximab as a background, and they played with the ability of the cell making the rituximab to alter the glycosylation. And so the first three, you see the red boxes on the left. The second line down, you've got sialylated antibody, which has a slightly different and reduced effectiveness compared to the parent rituximab. When you change that and you actually enhance the aminogalactose on the surface with the yellow circles there, and the third one down, you get a slight increase. The other thing that you can do is you can get rid of the fucosylation. And down here in the bottom, you see two different antibodies where they've gotten rid of the fucose on the first GalNAc in the glycosylation. And you can see that the potency and the binding to the target have gone up by over 100-fold when you got rid of that fucose. So that there are clearly advantages to getting rid of that, and you get a much different kind of antibody as a result. And in this setting, what they were able to show, for example, is the amount of effectiveness, the binding in an ELISA, and the effectiveness of rituximab ADCC was markedly enhanced when you got rid of that fucose in the green and in the light orange line that you see there. It's important to think about resistance mechanisms whenever we talk about cancer therapeutics. And so it's important to just consider what the possibilities are. The first is that antigen heterogeneity or mutation can result in a loss of a target for the antibody. And the same is true if you downregulate the antigen or change its expression. The pharmacokinetics can be important. Antibody stability, filtration by the kidney, and so forth are part of that. You have vascular permeability issues, tumor interstitial pressure issues, antibody size, and then, of course, affinity in terms of deciding how much you have to get to the tumor in order to have an ideal effect. You can also think about bypassing the signal transduction pathway, which is being targeted by the antibody, which gives you an absolute immunity. You can lose the ability to present antigens at the MHC and downregulates MHC expression, which gives you another effect. And so you can also get regulatory T cells in order to decrease that activity. So there are lots of ways that the antibodies can fail. And of course, we see that in that no antibody therapeutic is 100% effective. And in advanced disease, almost all of them eventually fail. So I just want to touch on antibody conjugates as as an example of how we can use antibodies, because these are one of the most common uses of antibody therapeutics today. And I just want to point out that you've got the antibody itself, and there are two examples here: trastuzumab on the right, for example, which is it's linked to the drug through a linker which is cleavable or non-cleavable depending on the way it's set up, and then you have up to uh, 3 to 4 warheads, which are the molecules of maytansine, which are linked to that antibody. And for the most part, you find that the absolute payload for most of these antibody conjugates tends to be between 3 and 4. It can be as low as 1, but it's hard to get more than 4 on without really interrupting the function of the antibody binding. And so, as you think about these targets, you have the ability to change the linker, you change the affinity of the antibody, you change the drug that's being carried, and all of those things will give you different properties. And so, by fine tuning all of the various properties in this little jigsaw puzzle you see here, that you have an opportunity then to really create very personalized, selective medicines for the treatment of specific cancers. And that didn't go forward. Just some factors that are important, including antigen selection. You want to make sure that you're having differential expression and potentially internalization. You can have the antigen presented in the HLA context for internal antigens. You usually want to consider IgG1 as your primary treatment because it supports ADCC. You want to consider the glycoform, and you want cysteine-cysteine bonds, which keep the antibody in its appropriate conformation. And where you place the conjugates on the antibody is a whole technology that there are lots of little biotech companies founded on various technologies to label these antibodies now. And I think suffice it to say, it is a specialty, and not all antibodies are labeled the same way. And depending on the labeling and the linker, you can end up with very different results. The warheads are mostly usually 4. Auristatin is the most common. You sometimes see calicheamicin, which are alkylating agents, minor groove binders, but the common ones are auristatin and maytansinoids. A recent approval in breast cancer using bavdegalutamide is a novel thing that is opening up additional opportunities, remembering that these are small molecules that can be exported through MDR and other pathways, and just because it gets in doesn't mean it stays in. The linkers, you want just the right affinity. You want it to come off appropriately, but not come off before it gets to the tumor. So the better the affinity, the more likely it is you're going to get the drug released in the appropriate site. You can also just conjugate radioactivity here and do a simple radiochemistry using zirconium for imaging or lutetium for therapy, and it gives you the opportunity to really use that antibody to bring the radioactive isotope to the target in a specific way. And then there are 2 other categories that we really don't have time to talk to today. One is the creation of bispecific T-cell engaging antibodies, which is a big growth field in the antibody space. And there are a variety of very successful looking antibody constructs using BiTEs against solid tumors. And there is one approved BiTE for looking at a lymphoma treatment. And then the other is the CAR T-cell strategy, which, as you know, the development of T cells and T cell based therapy is very much a growth industry in oncology today. And much of that is based on making chimeric antigen receptors in T cells by using single chains from cloned monoclonal antibodies. And so I think that what I want to leave you with is that monoclonal antibodies form the basis for multiple classes of new therapeutics. In each class of therapeutics, there are considerations to understand the properties of pharmacology and the mechanism of resistance, and all antibodies targeted at the same antigen are not the same. The antibody therapy itself is likely to be increasingly linked to T cell therapy, both through transfection of the T cells and by co-administration of antibodies and T cells in the therapeutic regimen, and that antibody-based therapeutics will continue to be developed for new and existing targets, with biosimilar products beginning to enter the market over the next several years. So I'm going to stop there and take any questions that people have. Thank you for your attention.
Well, we are back and live. So I'd like to thank Doctors Gainor and Spriggs for wonderful talks. And I'd like to remind the audience to please participate. If you have a question for Doctors Gainor or Spriggs, please enter it in the chat and we will ask your question. The first question that we have is for Dr. Gainor. And it's in a couple forms. So I'll sort of try and separate out a little bit. When it comes to upfront comprehensive genomic profiling in lung cancer with a positive PD-L1 score, it is suggested that we don't use EGFR or ALK and to wait for the GCP score. Does the same thought process exist with newer oncogenic drivers like MET exon 14, NTRK, RET, and I'll add BRAF because I'm a melanoma doctor and I know that there are mutations in BRAF in lung cancer too.
Yeah, it's a great question, and it's really clinically important because what ends up happening is the PD-L1 results will come back in a day or two. Patients anxious to get started on treatment, you see that there's a high PD-L1 score, and you're saying, no, let's wait for the comprehensive molecular testing. I do think it's still important to get that complete testing, especially in someone who has a high likelihood of a driver. And I would extend that beyond just EGFR and ALK. We know with MET, NTRK, ROS1, for the most part, TKIs plus PD-1, at least in lung cancer, don't mix well. BRAF, Ryan, I know you could point to that, that there is data of combining BRAF MEK plus PD-1, but generally, for most of these oncogenic drivers for which we have FDA-approved therapies, right now there are 7. Most of these patients, are those low TMB non-smokers. They're going to benefit most from targeted therapies. So I generally would recommend waiting for that full molecular testing results before starting therapy. Now, there are certainly clinical scenarios where you simply can't do that. You need to get started on therapy first. And in those situations, when I have a high likelihood of having an oncogenic driver that I can target, so these are never smokers or light smokers, in those situations, I'll actually start with chemotherapy alone and hold out on the PD-1 inhibitor. The reason I do this, and just to make the explicit point, is that the half-lives of PD-1 inhibitors are long, and so you don't want to be in a scenario where you've given the PD-1 inhibitor and then 2 weeks later a targetable mutation comes back. You want to add the TKI, and in that setting, you run into toxicity, and you really can't add it back for some time. So that's generally what I do in that situation.
So just to be clear, if you have a driver mutation and you're going on a specific TKI, not only are combination trials with TKIs and PD-1 inhibitors sort of too toxic to think about, but also sequencing from PD-1 inhibitor first and then going immediately to the TKI is also highly toxic?
Correct. Correct. In fact, with the prototypical drugs such as in the EGFR setting, starting a TKI within 3 months of receipt of a PD-1 inhibitor saw significantly increased toxicity in a retrospective series.
Got it. Thanks for clarifying my question. Dr. Spriggs, I have a question for you. And again, I want to remind the audience, please send in as many questions as you have. We have 10 more minutes and I can ask questions quickly. So one of the things that I'm always interested about in terms of antibody drug conjugates, and you did a beautiful job of sort of describing the specificity of the specific antibodies and how they can be, it can have a higher binding capacity and through all the engineering that can be done. What can be done about increasing antigen expression? Sort of thinking about it sort of differently.
I think that's one of the intrinsic problems with the antibody conjugates is that it's hard to know what can be there aren't a lot of interventions that are commonly offered there. What you end up with is a selection pressure that may eliminate the expression of the antigen you're interested in. Now, that's not necessarily a bad thing if the antigen is chosen well, which is to say that if you look at MUC16, which was an antigen that was commonly targeted in the context of ovarian cancer, and Genentech built an ADC in the mid 2016-17 range, and they would decrease the expression, and the CA125 might fall, but it was not persistent. And but the MUC16 also accelerates the growth of ovarian cancer, and so by choosing an antigen like that, the MUC16 negatives kinds of tumors that result from that selection pressure actually grow more slowly and are advantageous. So what the next generation of these kinds of strategies is going to be to engineer antibodies that have on one arm of the Y, if you will, you've got one antigen, and on the other arm of the Y, you've got a separate antigen. So you then have two antigens that you can get to deliver the warhead, and that gives you twice as many chances to eliminate that particular cancer cell. If you're looking at one of the more novel antibodies are antibodies that are in the context of HLA, and so you can actually make antibodies to the HLA with a specific peptide in the presentation cleft. And those there's another mechanism of escape where they lose the HLA expression, and therefore the antigen doesn't get presented. And then you have to start looking for ways to increase HLA expression, and there are strategies. Some of the interferon strategies may have an effect there, but by and large, it's one of the limitations of the technology.
Got it. And a follow-up to that actually comes from Bruce Chabner, who's, I think the first part of his question is, do you know about resistance to ADCs? You certainly described a few mechanisms. The follow-up was that, is there antigen shedding or resistance to the payload specifically been described?
Yeah. So the antigen shedding that's a matter of choosing the right target. And for example, the Genentech target that product I was talking about was shed, and it was the CA125 target, which we all know circulates and is in the serum. And so you have to go further down on the MUC16 in order to find components of the material that are not present in order to avoid that issue. But antigen shedding is a component of what can decrease the effectiveness. But because of the we're studying a new generation of the MUC16 targeting, looking at the protein stub that's left after the CA125 cleavage has released CA125 into the extracellular fluid. And there, what we're seeing is that there's a strong association between sensitivity to a maytansine-based ADC and the expression of MDR in the cell lines that we're looking at. So there's quite clearly an opportunity to escape in that mechanism as well. And so if you're looking at tubulin products or govinotecan and the camptothecin products, those are clearly substrates for MDR, and again, better technology is needed.
Awesome, thank you. And Dr. Gainor, question for you. Can you comment at all about combination therapies in the setting of other molecular subtypes like KRAS mutant or TP53 mutations, and thinking about how to incorporate IO in perhaps targeted therapies that are targeting, say, G12C, and then any other kind of things targeting these types of mutations?
Yeah, so I think KRAS is one of those molecular targets. One, it's exciting that, first we've known about KRAS for so long, and now we actually finally have drugs that are able to directly block KRAS if you have the KRAS G12C mutation, which actually accounts for about 40% of all KRAS mutations in lung cancer. So that's exciting. The second piece of that is in contrast to some of the other molecular targets I mentioned, like EGFR or ALK, G12C is enriched among smokers. So these are higher TMB tumors. These are likely to be more immunogenic tumors. There's some preclinical data suggesting one of the new G12C inhibitors can increase immune infiltration in preclinical models. I do think some of the same issue, and some have argued, well, the G12C inhibitors are so selective that we're not going to run into issues. But I have concerns that G12C plus PD-1, there's still potential for toxicity there based upon all of our prior experience. So I think there's more of a rationale for either some additive benefit, but I think we have to be quite cautious and not run into it too quickly and actually do the formal studies.
But presumably those studies are being designed or launched.
Yeah. Those studies are actually ongoing right now. So with both sotorasib and adagrasib, there are cohorts evaluating combinations with PD-1 pathway blockade. I think it's going to be needed to do that because the monotherapy activity of the G12C inhibitors, while good, isn't going to be enough to displace first-line therapy without either some combination or within a subgroup of KRAS.
Got it. Thank you. And Dr. Spriggs, a question for you. When thinking about engineering new types of ADCs, you had mentioned towards the end of your talk about T-cell engagers. And is there any thought about selecting a payload that may lead to immunologic cell death, and then also be able to engage with T cells to not only sort of have an agent that does that, but then help encourage the immune system to be more effective?
Yeah, that would be an unusual development. I think that the simple bispecifics or their variants, either the diabodies or other simple molecules, are much more common. There would be the opportunity to put. Remember that the it doesn't have the full tail, which is typically the FC region is where you're attaching your payloads. But that's a simple protein engineering problem, and I am sure that can be handled. But I've not seen a lot of interest in that yet. I think people are still uncertain about whether or not the BiTEs are going to really find a home in the context of the solid tumors because of the very immunosuppressive tumor microenvironment. And so the real question is whether or not we can overcome the tumor microenvironment. And for the bites, are there going to be T cells there to engage and to actually take part in the killing? And I think that's in the solid tumor space, that's yet to be determined.
Yeah, I would say one thing to that is that we now have a tumor that's generally thought to be relatively immunologically cold in uveal melanoma, where a bispecific molecule with T cell engager has shown to be effective. So there may be more T cells that are there than we might think.
And the ones that get there may not be able to leave. So
yeah, exactly. I think we have time for one or two more questions. So one question for you, Dr. Gainor, is there more efficient means to test for PD-L1 outside of using FFPE? Can it only be done with IHC? Are there other panels that can be done? Is immunofluorescence being considered? If you could sort of talk a little bit about maybe next generation, or there is no next generation of PD-L1 staining.
Well, I think for PD-L1, it's there have been if we would have been talking 5 years ago, we would have talked about all the various different clones and things like that. Thankfully, there have been efforts to harmonize scoring and things like that. So I think we have a pretty good understanding of the concordance of the various assays. But I think to your point about next generation assays, it's less so about PD-L1 alone and more about evaluating other components of the immune system. So, immunofluorescence is one way to look at multiple markers simultaneously. And that's something we're doing here at Mass General, looking at, it requires one slide and you can look at, 6 different markers. Obviously, to bring that into a clinical test is the order of magnitude higher bar to do, but I think it gives a lot more richness to the information of what's going on in the tumor microenvironment and the relationship of different components of the immune system and markers, as well as checkpoints.
Awesome, thank you. And another question came in for you, Dr. Spriggs. If a bispecific antibody is beneficial, how is that better than a cocktail of antibodies against multiple possible target antigens?
Yeah, and I think that's an interesting question, and it brings up: are we doing cocktails of antibodies for delivery? And in the current American system, one of the things that means is in order to do that, you either have to have an agreement among 2 companies or you have to have antibodies that are owned by the same company. And the anti-CTLA-4 PD-1 antagonist combinations is a good example of that, although it's in the immune checkpoint space. And so the by and large, that's not being done because of the developmental difficulties in terms of, is there a path forward to actually getting an approval from the FDA for these kinds of cocktails, which ones are active, which ones aren't. And so I think that cocktails of antibodies have eluded us in part because of the way our healthcare system is structured. But I think that there is no reason not to think that. I think one of the other spaces that would be interesting is to use CAR T cells against 2 different targets, which would be another strategy in the same direction. And my colleague, Dr. Oladapo Yeku, has been doing some of that in the laboratory, and it's actually an interesting thing, and you get a substantial increase in delivery. And so one of the things that we're looking at now is trying to overcome the payload limitations of CAR T cells, where you can really only put 1 or 2 additional proteins into the system. And we have an artificial human chromosome strategy, which would allow us to deliver 10 or 12 different cytokines and single chain antibodies simultaneously using the T cell basically as a vector to deliver it to the tumor site. And so I think there's excitement in that space as well.
That sounds awesome. Well, our hour has concluded. I'd like to thank Drs. Gainor and Spriggs for wonderful presentations and for this really stimulating discussion. To finish this this session, I'd like to welcome Dr. Chabner to say a few words to close this out.
Thank you very much, Ryan and David and Justin, for marvelous talks. I hope our audience has gotten a sense of the rapid evolution of therapy, particularly related to the use of monoclonal antibodies in all sorts of novel ways. The other thing that occurs to me is that it's really going to require an optimum therapy and integration of multiple strategies, and one of those is to preserve the role of chemotherapy. We've seen that with the checkpoint inhibitors in lung cancer, as Justin explained, and also with the use of ADCs, which use chemotherapy molecules as their payload. I think it's really just the principle that's important here, and that is that all modes of therapy will be necessary in order to get optimum results, and integrating these will require very careful but intelligently designed trials. So I thank the audience for being with us for the series of talks in translational oncology, and thank our sponsors.